So other variations that we can see when talking about the nitrogenous bases and the nucleotides themselves is in modifications that can occur after we synthesize DNA. So these are post modifications where we are taking a group that already exists, for example, cytosine, and modifying the chemistry of the cytosine to make a different nitrogenous base. So, for example, 5-methylcytosine is common in eukaryotes, and I'll show you an image of that. This is a varied cytosine where a methyl has been added to the 5th position. And we also see it in bacteria. There's also a N6-methyladenosine, meaning that on the 6th nitrogen of an adenosine, we have added a methyl. Also found in bacteria, but not in eukaryotes. So this is, for example, if you found this in a eukaryote, that means that it had to be produced from bacteria, and it's not actually naturally being produced from a eukaryote. These types of modifications, for these adding a methyl group, for example, to a cytosine or an adenosine, and making these varied groups are called epigenetic markers, where a way to mark DNA so that cells can degrade foreign DNA. So sometimes our DNA will be tagged with these markers, these methyl groups on the nitrogenous base, again, as a way for our body to recognize, hey, that is foreign DNA that comes from a bacteria or a prokaryote, and it needs to be degraded. Epigenetic markers are often also seen occurring in relation to diseases often. So this can be used as a way to tell your body if a gene should be active or not. So eukaryotes like us use this tagging of methyls onto a nitrogenous base to turn genes on and off very often. When the tag is there, often we'll turn the gene off. Sometimes it does turn it on, but often it turns it off because you're essentially blocking that gene from being normally accessed by putting the methyl group on there. So sometimes an environmental stimulus or food we eat or just chemicals that we encounter can actually epigenetically modify our DNA and accidentally turn on or off different genes, which can then be the cause of disease sometimes. So here's just to visualize that, some of those variations to different types of nitrogenous bases. We can see 5-methylcytidine, which I gave as an example. We're on the 5 position. Again, we have a methyl that's been added. This is the N6-methyladenosine, again on our 6th place. On the nitrogen part, we have a methyl that is added. A similar one here on guanosine, where we've added a methyl onto the nitrogen of the 2 position. And lastly, this hydroxymethylcytidine. So similar to our methylcytidine, also in the 5 position, but we have a whole CH2OH group that has been added. So some other nucleosides that are modifications we see are variations, whole entire variations of nucleosides that are found. And these are often found in RNA as opposed to DNA. One of those groups is called inosine, which is found in what we call the wobble position. We'll talk more about that in tRNA, transfer RNA. So inosine is a whole new nucleotide. It's a different nitrogenous base that we sometimes see encoded in RNA in position of where adenosines normally actually tend to be. There's also another nitrogenous base that is found called pseudouridine. So this is kind of a base that looks similar to uridine, found again commonly in tRNA and also in ribosomal rRNA. It's also found more commonly in eukaryotes than in bacteria, even in bacteria that is symbiotic with us. The way that it is made, this nitrogenous base, is from taking uridine and then isomerizing it after an RNA synthesis. It's thought that this pseudouridine formation comes about because it helps stabilize tRNA better and also might help with the folding of rRNA. So this is why we think it has developed in these places within tRNA and rRNA. So here's some of those groups. These are additional groups, again, that are found in especially RNA, types of tRNA and rRNA, being utilized as unique nitrogenous bases as a part of a nucleic acid or nucleotide. So here's inosine, pseudouridine, and two others that we did not talk about. But again, these are similar small modifications. You can see thiouridine. This uses a sulfur. So it looks like uridine, but with a sulfur group on it. And 7-methylguanosine. This is a guanosine that actually has a methyl group on the 7th position and very uniquely has a charge on it, which you'll see none of these other nitrogenous bases are charged.